Hello, everyone. Welcome to week number 5 in Biology 120. This is one of our weeks on genetics, which of course I love, so I hope you do too. Let's see what we've got going on. So last week we learned about metabolism. So metabolism is all the biochemistry that happens in cells. And that refers to everything that has to do with breaking down food and making all the molecules that you need. Every kind of chemical reaction, all those put together are called metabolism. And we learned about cellular respiration, and that's the way that organisms get ATP, make ATP, adenosine triphosphate, an energy molecule, from foods in different ways. Most of the time by the use of oxygen, but there are a few processes that do not use oxygen, like anaerobic respiration and also fermentation. So we learned about cellular respiration, and you also learned about photosynthesis, which is those organisms on the planet that can take sunlight energy and convert it into glucose. And that's very important. That's the whole basis of life on the planet, because all of us, no matter what kind of organism we are, depend on those autotrophs that produce their own food, glucose, through the use of sunlight energy. Okay, so that's what we learned about last week. Like I said, we're moving on into genetics this week. There are 3 really heavy chapters in your textbook, and so I am suggesting that you skim through the chapters 10, 14, and 15 about the structure of DNA, about how DNA replicates, about how DNA is used. So of course, everybody knows that DNA is the molecule of heredity by now. All of y'all know that. But why is it so important? Because it contains all the instructions to make all the enzymes that your cells need to make all the different types of cells that you have and to run all the different types of cells that you have. That's the importance of DNA. All the instructions are there to make the enzymes and make the cells that make you the organism that you are. So that's the job of DNA. The job of DNA is not to replicate or make copies of itself, but of course it has to do that if the organism is going to reproduce, right? And repair cells and all that sort of thing. So skim through those chapters 10, 14, and 15 in your OpenStax textbook. At least open them and look at the pictures and read the little extra articles because they're very interesting. And one of the, oh no, I forgot which chapter. I just saw it. It's either 14 or 15. There's a little blurb about forensic science, and I'm going to cover a little bit about that today. Read your Module 5 overview. It's fairly short. And watch the videos. The videos are good and they will help you. All right, then you've got discussion board postings this week and a milestone to complete. So let's talk about the discussion board postings for a second. The question is, if you were having this discussion with your friend about cancer, now everybody has heard of cancer. So what the heck is cancer? Cancer is not something from the outside. Cancer is some of your cells that are dividing out of control. And the most important characteristic of cancer cells is that they metastasize, and that means that they move from the original location to other locations and set up shop and start dividing and take up all your nutrients. Yeah, that's the problem with cancer. All of us have been affected by it in one way or another. So you're having this discussion with your friend about cancer and, well, what really is the cause of it? Is it a problem with cell division? That's mitosis, or is it a genetic disease? And that means in the genes, so a gene is a little portion of DNA. So is there some gene in cancer cells that turns on or turns off that turns it into a cancer cell to make it divide out of control? This is your question. Now, in this description, it says or both. Now, what did I tell you last week? If it says or both, you know it's or both. Otherwise, it wouldn't be mentioned. That's just a clue about how to read questions like this. So let's talk about this for a second. Over on the side over here, I have the cell cycle drawn out like I would want to draw it out. Not like I always see it in textbooks. But anyway, so let's start like at 12 o'clock, right? That's like at the beginning moment of the life cycle of a cell. And that's what the cell cycle is. It's the life cycle of a cell. What happens to it over its lifetime, right? So we start out and the cell goes into something called G1 phase. Now I know I have G0 up there. It goes into G1, and this is just general growth of the cell. So if you're a eukaryotic cell, your mitochondria reproduce, you're making more ribosomes, you're making more endoplasmic reticulum, et cetera, et cetera, et cetera, lots of new enzymes. General growth is in the G1 phase. All right, I'm going to come back to G0 in just a second. Then, when the cell has grown enough, it phases out into the S phase. S means synthesis. And so what is synthesis? That's making something. You're synthesizing something. You're making DNA in this phase of the cell cycle. So you're replicating your DNA in the S phase of the cell cycle. Then you move into the G2 phase. So G2 is where all the cell machinery is produced, where that's necessary for cell division to take place, like the centrioles, like the spindle fibers. Now, if you remember hearing about this in some other biology class, that's what these are. These are parts of the machinery in the cell that are necessary in order to make the cell divide. Okay, that's what happens in G2. And then we have M, which is mitosis. So that's a division of the chromosomes in the nucleus. And C is cytokinesis, division of every other part of the cell besides the nucleus. All right, so that makes sense. So I've described the cell cycle except for that G0. So the job of a cell is not to reproduce. Every cell has work to do. And that's what G0 is. So the cell starts out in G1 and it grows to a certain size and then it stops, and it stops in G0. And it does its work. If it's a heart muscle cell, then it pumps and pumps and pumps. If it's a cell of your small intestine, it takes in nutrients. If it's a nerve, it conducts electrical impulses. So whatever the job of that cell is, that's what it's doing in G0. If it gets stimulated, then, if there's harm to the cell, if there's damage to the cell, if it's just a cell that wears out, it'll move on into G1 further and grow to a bigger size, then S, where the DNA is replicated, then G2, got all that cellular machinery being put in place, and then the cell divides, okay? So that's how the cell cycle works. Now, here's the problem. With cancer cells, they just keep going around the cell cycle and they don't stop. The other important thing is that there are checkpoints all along this way. So if the cell hasn't grown enough in size in G1, there are checkpoints there where the cell checks itself to make sure everything is in place before it moves on to the S phase. Cancer cells ignore the checkpoints. They don't care. They don't care if the cell's grown enough. They don't care if the DNA's been replicated correctly. They don't care if the cellular machinery is there to make cell division happen. And so you get all these kind of wonky looking cells when you look at cancer cells. Some of them are gigantic because they never actually split in half. You get all kinds of strange things happening in cancer cells because they don't observe all the rules in the cell cycle. So that's cell division. Well, what about genetic diseases? Diseases of the genes. There are certain genes in cells. There are tumor suppressor genes, and there are proto-oncogenes. So proto means before, right? So an oncogene means a cancer gene. So a proto-oncogene means a cancer gene before it's a cancer gene. So if proto-oncogenes are mutated, then they can turn into oncogenes and make a cancer cell. Tumor suppressor genes prevent tumors from forming, right? There are stops in place. There are processes in place that will take a damaged cell and kill it off by a process called apoptosis. And it's not apoptosis, it's apoptosis. There are processes in place where a cell can undergo cell suicide instead of becoming a cancer cell. But if you have problems with a tumor suppressor gene, it gets mutated and then it doesn't work, you can have a cancer gene. So this is the type of information that you're going to be looking up in chapters 10, 14, and 15 to help you answer this question about whether cancer is an issue with cell division and mitosis, or is it a genetic disease in the genes, or is it both? And you're also going to discuss how you can change, anyone can change their lifestyle in order to lower their cancer risk. Now, one thing everyone should understand, there is nothing that you can do to completely lower and prevent getting cancer. There just isn't. If you live long enough, your chance of developing cancer goes up, because just living life out in the world is going to increase your chance of cancer. So realize that when you're talking about this. But are there lifestyle changes that you can make that help you lower your risk of cancer? In your response post, you're going to talk about environmental carcinogens. A carcinogen is anything, any type of mutagen, anything that changes the DNA, that's a mutagen, that causes cancer. So there are all kinds of environmental carcinogens. And you know some of these. How do you get skin cancer? You're exposed to sunlight. What's in the sunlight? Ultraviolet light, right? So you know things like that. So you know environmental carcinogens, and look up some more, because there are all kinds. And we're exposed to so many different kinds. Though I think fewer these days than way back in the 50s, 60s, and 70s for sure. And how can we get rid of these environmental carcinogens from the environment so that we're not exposed to them anymore? All right, so those are your response posts. Remember, the initial post is due by Thursday, response post due by Sunday. Then you got your milestone 3 of your final project. So this is the third section that you're working on. Follow the outline that you're given. This is what you got to cover. Methods of conservation for your topic now, right? So go back to your topic, whatever it is. Global warming, eutrophication, water pollution, microplastic pollution, whatever it is. Methods of conservation. So think of this section as prevention. How could you prevent, how could you take steps to prevent this thing from happening? Prevention. That's methods of conservation. Education. Why is education of the public important? Education of the public or users or companies or whoever. Why is education important in stopping this problem, preventing this problem, solving this problem? Okay? Solutions are the next section. These are the fixes. So you already got the problem. What can you do to fix this? What are you going to be able to do? How are you going to fix a lake that is going through eutrophication? For one thing, you're going to stop dumping fertilizer into the water. Okay? That's one fix right there. Ethical considerations. So are there any laws on the books or regulations that have to do with this? Are there any movements to change people's attitudes on this? What are the ethical considerations? Should we be trying to fix this problem? Or does it have benefits? And that comes in the next part, positive, negative aspects of the issue. Okay, because we've identified this as a topic, as an issue in a biology class, you know that it has negative aspects. Global warming. We're all going to be underwater if we don't stop this. And there are so many other negative aspects. But you also need to consider the positive aspects, because there must be some reason that this thing is going on, right? Somebody must be benefiting from this. There have to be some kind of positive aspects to this, because, excuse me, There's a reason that this is happening. And we always have to consider this. So, for example, if we're talking about global warming that's being caused by the burning of fossil fuels, isn't the production and sale of fossil fuels just a gigantic industry in the world? Doesn't this make people lots and lots and lots of money? Companies, but also people. Yeah, people are part of companies. Isn't this part of the problem? How are we going to change that? If we don't change the financial part of it, we are never going to fix global warming, I'll tell you that. And then what's the personal impact? What is the impact of this particular topic on you? Some of you have chosen really focused topics in your area where you live, and I love that, because then you really do have a personal connection to this topic. So tell us how it impacts you in this section. Include your citations and references. You're going to make slides this time. Label all of your sections. Methods of conservation up at the top of the slide. Education at the top of the slide. Label everything. This is not an English essay. And I've got to know that you know what you're talking about here. If you just run everything together and I have to pick and choose what I think you're talking about, I may not really connect with what you're saying. So please label all the sections. All right, the next section is extra because it's one of my great loves. DNA. Yeah. So I started graduate school in 1988, which is crazy. 1988, and this is when DNA fingerprinting was just coming about and just being talked about, right? So now we call it profiling. It's got so many names now. The name of it started out as DNA fingerprinting because of Sir Alec Jeffreys, and here's a picture of him right here. He was a genetics professor in England during this time. I mean, he's still alive, but he's retired now. So he was studying DNA, and he was studying the DNA in families. And he wanted to know if he could differentiate between members of a family based on their DNA and what their DNA looked like in a picture. And the picture on the right there is an example of something that he would have looked at. And everybody says, well, it looks like a barcode, like on the back of your groceries. And that's true. That's what it used to look like. It doesn't look like that anymore. So he developed this technique, and as he described it to his wife, she said, oh, that sounds kind of like fingerprinting, and hence the name DNA fingerprinting. So he developed this technique. It's called RFLP, and it's long and complicated, and that's what I used to do. It requires a lot of DNA that's not degraded, and it takes a long time to perform. It takes at least a week to do this whole process. And if anything's wrong with a sample, it just gets to be a huge mess on your radiogram, which is your X-ray film that you develop. But we could use it even back then. In 1985, police departments in England heard about Alec Jeffreys, and they had 3 murders on their hands, and they didn't know what to do. They had 3 young girls, teenage girls, who had been raped and murdered, and they had no idea who the perpetrator was. And the girls were in different towns, villages. So they went to Alec Jeffreys and they said, do you think that you could use this technique to help us discover who this perpetrator is? And he said, yeah, I'm pretty sure I can. So he said, all you need to do is get me blood from the men. So in England, they don't have the same civil rights that we do. In England, they just said, okay, all the men in the villages line up. We're taking blood from you. And so they collected over 5,500 blood samples, and Alec Jeffries started testing them against the DNA samples from the girls who were murdered. And during this time, they had someone who confessed. And Alec Jeffries ran the profile of this person, and he went back to the police, and I said, well, I have good news and I have bad news. I got a really good profile from this person, but his DNA does not match the evidence left behind in the scenes at the scenes of the crime. So he kept testing. Over 5,500 blood samples. That's a heck of a lot of blood samples to run, especially with this technique. Now, there was a man who convinced his friend to go give blood twice so that he didn't have to give blood. And he made some excuse about it. Well, his friend went to the pub that night, and after he'd been drinking, confessed to the whole group of friends that he had gone and given a blood sample and used Colin Pitchfork's, I'm not kidding, that's his name, Colin Pitchfork's ID. So he gave blood instead of Colin Pitchfork. Well, somebody in the group went to the police and said, there's something funny going on here, because why would this guy ask his friend to give a blood sample? So the police went to Colin Pitchfork. He was a family man, had a good job, never been in trouble with the law. Took a blood sample from him, and sure enough, his DNA matched those 3 girls, and he was convicted of their murders and is still in jail in England today. So that was the first case of DNA fingerprinting, but the very first case of DNA fingerprinting exonerated someone who was innocent. So that's kind of interesting. But then it was used to convict Colin Pitchfork. In 1988, and that's when I started graduate school, the FBI began using DNA fingerprinting. And this was before any states had DNA labs. Then, of course, after the FBI gets started, then the bigger states followed, and they developed DNA labs, and now DNA labs are everywhere. They're state labs, they're local labs, they're private labs, they're labs everywhere. And now the technique is much, much faster. You can use much more degraded DNA, which is awesome, because as you can imagine, at the scene of a crime, DNA is going to be degraded most likely. So there are brand new techniques. There are new techniques that are coming where you'll be able to analyze DNA out in the field. And that literally is right around the corner. And then the newest thing that's been happening with DNA and solving crimes is the use of genealogy to help solve crimes. And this is called forensic genealogy. So you're using websites like Ancestry.com, etc. There's so many more, 23andMe, all of those, to help catch criminals. And the most famous case, the first case to ever be solved using forensic genealogy, was the Golden State Killer, Joseph DeAngelo, who got away with murder for over 40 years. Now he's been caught, now he's in jail. But he was only caught through the use of genealogy searches. So looking back through family trees and looking at the DNA of related people in order to figure out who a criminal might be. And then you go and test that person, whether you get a sample from them from the garbage or from a restaurant or wherever, or you subpoena a sample from them and get one. This is how criminals are being caught these days, and this kind of technique is just going to move forward. I promise you that, because it's so individualized, right? Your DNA is specific to you and no one else on earth. And there are even changes in the DNA between identical twins. You think, well, identical twins are born with exactly the same DNA, which they are. But over time, even that changes. There are tiny little changes, but still there are some changes in their DNA. So this is a very, very important technique that's going to help us in the future, and who knows how many applications we'll think of in the near future even. So I'm really interested in this. If you want to talk to me about it or send me something, send me a case or ask me a question. I love this stuff. So ask away. I'm ready to jump right into the discussion. And with that being said, I hope that you have a wonderful week this week number 5. So we're getting past the midpoint here, and we're jumping into genetics. So let me know if you need anything.